Hello everybody, I welcome you all to today's video. Are you somebody who is really curious to know what are the instruments that's used in bioscience research? Well then this video is definitely for you. Today we are going to discuss the top 10 instruments you must know if you want to get into biotech research. I'm Dr. Vaishali, academic specialist at Biotechnica. Biotechnica is a space where we discuss anything and everything regarding your bioscience career. And come, let's explore the topic. The first instrument that we're going to talk about is the electrophoresis unit. Now, electrophoresis unit is used for separating the macromolecules in a fluid or gel. Now, what are these macromolecules? It can be proteins or plasmids, RNA, DNA, or even nucleic acids. Now, how are they separated, right? So, they are separated based on their charge or binding affinity and their size under an electric field. So, here what happens is you have two different things, that is the sample and the gel and also the electrophoresis unit. So, here first you go ahead and you prepare your sample. So in the sample, you will also be adding a dye, which will later be used for visualizing, right? And then you prepare the gel. And after that, the gel is being casted in the electrophoresis unit, which you can see here in the figure. So once this gel is casted, then you go ahead for sample loading. So these samples are loaded in the wells that is provided in the gel. So after loading these wells, then the process of electrophoresis starts. That is, the positive and the negative electrodes are fixed and the charge is being started. So when that happens, then the sample that has been loaded in the gel, it starts moving along the electric field, and according to their charge, binding affinity and size, these macromolecules will be separated along the gel as they move. Now, after the gel electrophoresis is completed, then the next step is to observe the gel under the UV light, which we are going to see in our next slide. That is the UV transilluminator. So here what we do with this particular instrument is that it helps us to visualize the gel, the electrophoresis gel that we've obtained after separating these macromolecules. So it's basically to visualize the target DNA or RNA which have been separated by electrophoresis. Now, the gel are stained, right? The gel is stained as well as it is passed through the UV light. So when it's passed through the UV light, the stained particles in the gel, that is the macromolecules that are stained, will be illuminated, and it will be and we'll be able to observe it with our naked eyes. Now, what are the uses of this UV transilluminator is one, if we have to see what are the PCR products that has come up, right? Second is enzyme digestion. That is, if you have used restriction enzymes to digest your DNA and you've run it through the along the electrophoresis gel, then those products also you can observe under UV transilluminator. Third is to quantify the DNA, and fourth is to verify the RNA integrity after the RNA extraction process. If you have to see if the RNA that you've obtained is integrated or not, then yes, then UV transilluminator can be used for this process as well. Now, the third instrument that we're going to see is nothing but PCR. PCR stands for polymerase chain reaction. So here we use PCR mainly to amplify the segments of DNA or create multiple copies of one DNA product. So that is why we basically use PCR. Now in PCR, we use different components such as Taq polymerase and DNA primers. Now, Taq polymerase is a thermostable DNA polymerase that is used for creating replicates of DNA, that is for synthesizing new DNA strands. So that is why we use Taq polymerase. So this particular Taq polymerase is obtained from Thermus aquaticus. So this particular polymerase is going to be thermostable. So it's going to be intact even at higher temperatures. So that's the reason we use this particular DNA polymerase. Second, we also use primers, because primers are nothing but the initiation or the starting point for any replication or formation of new DNA strands. So yes, for even polymerase to act or to start its activity, DNA primers are necessary. Now, what is this PCR? Now, PCR is going to be a repeatedly cycled process. That is, the cycles in the PCR are going to keep repeating so that we obtain more and more strands. That because our aim here to amplify the segments of DNA, right? Now, how are we going to repeat the cycle? Majorly by temperature changes. Now, let's now see what are these temperature changes and what are the steps involved in PCR. So the first step is denaturation. So we know that the DNA is double-stranded, right? For the first step that we do in PCR is to convert it into single strands, right? Because for replication to occur, we need the DNA in single strands. So that is denaturation, which occurs at 96 degrees Celsius. Second is annealing. Now, annealing is a process where the primers come and attach to the single-stranded DNA, right? So only when these primers are attached, which is nothing but the starting point for our polymerase to act, only after that our polymerase will come and act, and this process will start to occur. So that is annealing, which happens at 55 to 65 degrees Celsius. The third step is extension. Extension is nothing but the activity of Taq polymerase. So the Taq polymerase comes and attaches itself to the strand, and it basically keeps attaching these new DNTPs, and it forms a new DNA strand in itself. So this is your new DNA strand which has been formed by Taq polymerase. So these are the three different steps in PCR which happens inside this instrument, and it is controlled by the changes in temperature. Now, the next set of instruments that we are going to see is the microbiological instruments. So first we are going to start with a microscope. So microscope is an instrument that is used to magnify very small objects, especially the objects that cannot be seen with naked eyes. So it's majorly used for observing microorganisms and their features. Now, what are the different types of microscopes that's available? First is the compound microscope, which has two sets of lens, that is objective and the ocular lens, and it uses the visible light for its for its instrument. The second is the dark field microscope, which basically uses the illumination that is produced from this instrument as its source of light, and because of this, what happens is the specimen will be observed as white in color with a dark background. So that is the reason it's called a dark field microscope. Third is electron microscope, where instead of using light, electrons are used as the source for observing the specimens under the microscope. So electron microscope can be of two different types. One is SEM, that is scanning electron microscope, and TEM, that is transmission electron microscope. Next is fluorescence microscope, where fluorescence is used for observing the specimens, and the last is the contrast or phase microscope, where the difference in refractive index is used as the principal. Now, the next instrument we are going to see in microbiology is the autoclave. So autoclave is a physical method of sterilization where heat is used for sterilizing the medium or the vessels, etc. So it's basically used for sterilizing bacteria, viruses, and even spores, right? So it is also called as a steam sterilizer because steam is used as the basic mode for sterilization. So generally it is used at 121 degrees Celsius for 30 minutes with 15 psi of pressure. Now, what are the different types of autoclave that is available? The first is the pressure cooker type, which is nothing but the laboratory bench type autoclave that is used, which is a pressure cooker type. So here the autoclave of pressure cooker type. It comes with a vessel with a safety valve as well as the pressure releasing unit as well. So this is the N-type. It's also called the N-type autoclave. The second is the gravity displacement type, which is observed here in the image as well. So here the steam is generated inside the autoclave machine because of the water that is present, and the steam keeps helping in sterilizing the medium that is there inside. Next is the B type, or the positive pressure displacement. So here it has a different steam generator as one of the unit, steam generator. So in this steam generator, the steam is generated and this steam is then inflowed into the sterilizer where the sterilization actually happens. The fourth is the negative pressure displacement, also called as the S type. So here it has two components. One is the steam generator and the second is the vacuum generator. Right, so vacuum generator. So here what happens is, first the sterilization unit has been vacuumed out using the vacuum generator, and then the steam is inputted using the steam generator. So this is about autoclave. The third instrument that we are going to see in microbiology is the laminar airflow hood. So this hood is mainly used to prevent contamination at your workplace, especially when you're working with microbiological components. So here what happens is airflow is there with a uniform velocity. So the air is always flowing through this particular hood, which has a uniform velocity, especially in straight lines, right? So this airflow hood, before even the air enters, there are filters that has been placed to capture all types of impurities. Now, what are these filters that is used? One is a filter pad and one more is a HEPA filter. That's nothing but high efficiency particulate air filter. So these filters help in filtering all the impurities and then sending out sterilized air into the laminar airflow hood, right? So there are different types of this chamber. So first is the vertical laminar airflow chamber, which is very commonly used in the laboratories, where the airflow is vertical from the top to the bottom, right? The second is the horizontal laminar airflow, where the airflow is horizontal in direction. That is, it starts from one end where there are filters kept and then it proceeds to the other end. The third is the wall-to-floor airflow system, where the air comes from lateral sides of the hood, but it reaches the floor of the hood. So basically, the airflow will be inclined in direction. Now, the fifth instrument that we are going to talk about is the flow cytometry. So flow cytometry is used for analyzing single cells, right? So here, the lasers are being used for analyzing these single cells. So the laser could be single or multiple laser. So here what happens is there will be flow of cells like this. So there will be two types of cells. One would be stained cells, and one more would be non-stained cells. And the laser will be sent from here. So when the laser comes in contact with these cells, the stained cells will emit fluorescence, and all the cells, that is stained as well as unstained cells, will be emitting the light. So this particular difference is used to go ahead with analyzing these single cells in a cell culture, right? So it's helpful mainly for cell counting, for cell sorting, as well as used as a biomarker detection in protein engineering. And these cells that are used here, right, so these are suspended cells in fluid. So it's because there should be a constant flow of these cells along this particular tube. So obviously they have to be suspended in nature. Now, what are the applications of flow cytometry? First is for expression of cell surface and intracellular molecules. So this is one of the application. The second application is identifying the cell types within a mixed population. So these are just two of the applications that I have mentioned here. Now, the next instrument we are going to talk about is mass spectrometry. spectrometry. Now, here mass spectrometry uses the concept of mass to charge ratio for analyzing the macromolecules. So, majorly these mass spec is used for one is for identifying the unknown compounds, second for quantifying the known compounds, and third for detecting the structural and chemical properties of these macromolecules, right? So this is the instrument that is used in the case of mass spectrophotometry. So there are three different components in this mass spec. One is the ionization source. So here what it does is it ionizes the macromolecule into the gas ion phase, right? So that it will be helpful for detection. So this ionization source helps in converting the macromolecule into the gas phase ions. Second is the mass analyzer, where it basically separates and sorts all of these ions into based on the mass to charge ratio. So that is what a mass analyzer would do. The third component is the ion detection system, which helps in measuring these sorted charges, these sorted mass to charge ions, and they help in giving an output in the form of a graph. So this graph will help us in analyzing what kind of compound, if it is an unknown compound, what kind of compound it could be, or it also helps us in quantifying the known compounds. So these are the few of applications of mass spectrometry. Now, the seventh we are going to talk about is an incubator. Now, incubator is nothing but an enclosed and insulated device, right, which helps in growing and maintaining cultures, right? So, what are these cultures? These cultures could be microbial or they could be cell cultures as well. So, this enclosed space, as we can see in this figure as well, helps in maintaining and also growing these cultures. How does it do? Is basically by maintaining optimal temperature, humidity, carbon dioxide as well as oxygen. So these concentrations are varied according to what microbe and what cell culture is being used. Now, what the different types of incubator that is found is, first is the benchtop incubator, which is generally just controls the temperature alone. Right. The second is the CO2 incubator, where along with temperature, even the CO2 as well as the humidity is being controlled, right? And humidity. So these two are the additional features of a CO2 incubator, which is majorly used for cell culture experimentations. Next is the cooled incubator, where your culture requires a below optimum temperature. So here we use a refrigerated incubator, right? The next is the shaker incubator. Shaker incubator is an incubator that keeps, rotating, right? It gives a uniform agitation for the growth and for maintaining the cultures. So that is the use of shaker incubator. The last is the portable incubator, where the incubator is easily portable. So this is used for, field-wise, where you have to use the incubator in the field, like environmental microbes, or, where you have to use it in the field. So that is where the portable incubator comes handy. The next instrument we are going to talk about is the centrifuge. Now, centrifuge is used for separation of fluid, gas, or liquid. So it's based on the density. So what happens is, in a centrifuge, you keep the fluid inside a centrifuge tube, and these centrifuge tubes are placed inside the centrifuge. Now, this centrifuge will rotate in a very high speed. Now, because of this rotation, centrifugal force comes into action, and this centrifugal force helps in separating the molecules based on its density. So the highly dense molecule that is present in that centrifuge tube will come and settle at the bottom, and the supernatant is available on the top. This, for example, you can see it in case of cells or debris. So how do you separate the cells from its fluid or its medium? Is the broth specially is by centrifugation. So this will help the cells to settle at the bottom, whereas the supernatant will have the medium. Now, what are the different types of centrifuge that is found? So first is a small bench centrifuge that is majorly used in the bench level, right? You use it for erythrocytes or for yeast cells. Second is the large capacity refrigerated centrifuge. So as the name itself suggests, so it has a higher capacity than a small bench centrifuge, and it has refrigeration facility as well. The next is the high speed refrigerated centrifuge, where the speed of the rotation of the rotor can be changed as well. So there are different rotors that can be fixed to the centrifuge so that you have a variable speed of centrifugation. The last is the ultra centrifuge that is used for more delicate molecules, where you have to separate more delicate molecules in a higher speed and a very controlled environment. Now, after centrifuge, the next instrument we are going to talk about is the magnetic stirrer. So magnetic stirrer is used for stirring the molecule, basically for agitation as well. So it has a rotating magnet, or it can also have a stationary electromagnet that creates a rotating magnetic field. So basically we need a variable magnetic field on this particular instrument. Now, once we get a variable magnetic field inside the liquid sample that we have, where we want to separate the molecules because of stirring, so there you place something called a stir bar, which is here seen in this particular image as well. So you place this particular stir bar inside your fluid medium, and then you place this whole beaker on top of this stand. So once it is switched on, then there will be variable magnetic speed. Because of this variable magnetic speed, the magnetic bar, that is the stir bar that is inside the fluid, also keeps rotating. And because of this rotation, the fluid that is there inside the beaker also starts rotating, and this helps in separating the molecules. So there will be rapid movement and agitation in this particular beaker. Now, the last instrument that we are going to talk for today is the spectrophotometer. Now, spectrophotometer, it analyzes the amount of light that a sample can absorb, right? So in this particular image, so the sample which has to be detected will be kept in this particular slot that is there, and the UV or visible light, according to the type of spectrophotometer that you're using, will be passed through. So passing a light beam through this particular sample, and after that the light is then detected at the outside this particular sample. So this instrument has two different components. So first is the spectrometer, which helps in which helps basically in formation of the lights at different wavelengths, right? So because for different samples, the wavelength that has to be used in this particular instrument will be different. So yes, a spectrometer will help us in formation of these different wavelength lights. Second is a photometer that helps in detection of the outcoming light. So that is what a photometer does. So what are the different types of spectrophotometer based on the light that is being used? The first is the visible spectrophotometer, which uses the visible light. Second is the UV visible spectrophotometer, which uses UV and visible light. Third is the infrared. Fourth is fluorescence, and the last is the atomic absorption spectrophotometer. So these are the different types of spectrophotometers that are used for generating and for detecting the light that comes out of the sample. With this, we come to the end of this particular discussion. I'm sure it was super helpful. If you want more details on any more instruments, do let us know down in the comment section. Thank you so much, and see you all until next video.